Hi there and welcome to Kaleidoscope Live. I'm Tracy. I'm here with Dan, Joyce and with Sukhi, Shergill. And if you're observant, there is somebody missing. There is normally four of us here, but Dawn Albertson, apparently there's some bug going around at the moment and she may have caught it. So get well soon, Dawn, if only because I can't face doing this with just these two guys. We need you here to liven it up for us. We've got a couple of papers we want to talk about today. If you do want to put some questions in the chat box, it should be open and we will try and get through them. The links to the papers will be put up on the website. As you may know, if you've seen this before, we take this from the Kaleidoscope column that we do in the British Journal of Psychiatry, so the papers are also there. And I'm going to start off with a paper this week. It's a paper called Glycobiology and Schizophrenia. It's a review paper by Robert Mealer and colleagues from Harvard, and it's in molecular psychiatry, just out at the end of 2020. And the paper interested me for a couple of reasons. One is that it offers new insights, potentially, which we'll talk about, into what causes psychoses and what that might mean in terms of potentially new treatments. But I was also interested in the broader principle of what genetics work is beginning to do. And I wanted to talk about that part at the start. And I'm not a geneticist. Amongst the things I don't know a huge amount about, genetics is high on the list. But it's clearly important for us. So the things for me, the kind of breakdown in my head is always that if we think about any mental health problem, they are typically have a strong heritability. So that means that you can inherit it from your parents. And if I think about psychoses and about lots of them, it tends to be roughly 50-50. It varies. So some is your genes and some is your environment. And of course, your environment also impacts your genes, a thing called epigenetics. And when I was in school, it was very simple, what we would call Mendelian model that people might be familiar with. So if you remember A-level textbooks, you had things like your eye color or pea plants, and you had a gene that was dominant or recessive. And that doesn't really apply in mental health. In fact, it doesn't apply to much of healthcare at all, except for some very rare conditions. And more than that, it's the complex interaction of our genes. We've got thousands and thousands of them. So 20 odd years or so ago, they began to do, when I say they, people smarter than me or involved in genetics work, began to do things called GWAS studies, and that's genome-wide association studies. So what that does is it looks at your whole genome. It gets lots of people with maybe a condition that you're interested in and lots of people who don't have it, looks at their whole genome and begins to look for differences between people. And what we see with this is interesting. It confirms the fact that most mental health conditions are polygenetic. There's lots of genes involved. But what's really complex about this and what's confusing for me and lots of people is it can involve hundreds and potentially thousands of genes. And then we get all these really complicated questions. Does it matter which 100 or 1,000? Is it the combination? They each add a small amount to it? Does it depend that one gene might be really important if it comes with another one? And one of the challenges with GWAS studies has been trying to build, the associations between them are often quite weak. And what I think is interesting in more recent times has been a move to see that it's not just the gene that makes something, it's what might happen after that. So genes make proteins, they make parts of your cells, but there are other parts that are really important to that. We had a paper in Kaleidoscope, in fact, I think it's coming out in February, that's looking at what's the difference between a human brain and a chimpanzee brain. And ours is generally a lot bigger. But what that was interesting with that study was saying a lot of the genes are quite similar. It's the amount of times you make you get the gene expressed. So there's something about how genes are promoted. So that's, again, where there's maybe not a problem with the gene, but how you regulate it. And the other thing you can do with genes is or with proteins is you can change them after the protein's made. So I don't know what people's understanding of genetics is, but if you're kind of a bit basic like me, you've got transcription and translation. So you get the DNA, you read off it, and you make proteins. But there's a part called post-translational changes. So once you get little proteins in the cell, can you alter them? And the answer is yes, you can. And so that takes us on to this study and the process of what's called glycosylation or glycobiology. And what the authors say is interesting here. They looked at GWAS studies for psychoses. So they looked at these genome-wide association studies and what we could learn about people who have psychotic illnesses. And what they picked up that some of the really strong signaling is coming from genes that are involved in glycosylation. And what that means is adding sugars onto the proteins. So it's not the proteins you make in your cell, It's how you alter them afterwards. Now, that's interesting of itself. What we don't really know is, what does glycosylation do? So when you make proteins in the cell, what happens to them? And what happens if you add on the sugars? And there's some hypotheses about this. So glycosylation seems to affect how proteins are trafficked, to how they're moved around the cell, and maybe how they get to the cell surface. And what's also really interesting with this piece, which I think Sukhi's going to talk about in a few minutes, it goes back to some old culprits within psychoses, or suspects, of things involving dopamine, involving calcium channels and so forth. So on the one hand, it reaffirms part of what we know, which is probably helpful. It makes us have some confidence in the findings. But what's interesting for me is it's saying that there's something different about how these proteins are moved around cells that might be really important. And the authors say with that, as well as telling us potentially more about psychoses, and the other thing that it reaffirms to me is that we have a spectrum condition. This might be more important for some people, less important for others, is that it potentially offers new treatments. Our treatments to date tend to be about modulating dopamine pathways within the brain. There's interest in glutamate pathways. But here they're saying maybe what we need to do is getting within the cell and seeing how we alter those sugars. Is that something we could change if we needed to in someone who is having problems? So again, for me, I thought this paper was interesting when I saw it, because it helped me understand a bit more about the genetics of it and reminded me of the complexity of it. And again, maybe it helps us understand a bit more about what we might do from a treatment point of view. So what did you two think of this paper?
I mean, I thought it was a great paper. I think there are lots and lots of genetics papers, and the question really is, how do you get from showing that there are some genetic changes in one disease, how does that map onto people's functioning? So basically, in this paper, you say that some changes in genes that influence glycosylation, a process which is relevant to brain and neurotransmitters, is causing symptoms in patients with psychotic illness or schizophrenia. So how do you get from that gene change at a kind of group population level in 70,000 people that they might have looked at, to one person experiencing psychosis, either hearing voices or feeling persecuted, or even the kind of negative symptoms of that? So it's that mapping between the genes and the function of the brain, I think, which is what this paper is offering, a kind of intermediate, because what it's saying is the genes in themselves are kind of interesting and they're giving you a clue, but how do they change brain function to get to this issue that you have with changes in kind of cognitive performance and changes in your perceptual reality? And what this paper seems to be suggesting, that glycosylation is a process which may be contributing to that. And so the thing that was quite interesting for me is, well, how does that fit into the information that we already have about treatments and about responses? So all of the kind of current medications that we have all block dopamine receptors or modulate dopamine receptors in some way or another. And what this paper talks about is it identifies kind of 4 basic mechanisms that it thinks that glycosylation is involved with. So one of those is that it modulates the activity of these dopamine 2 receptors. So that is quite a nice touch, because what it does is provide a link between what we're doing therapeutically, which influences change in behavior and function, and links that to this process that these genes are involved with. So I kind of think for a long time that's been the bit of the puzzle that's been missing. The other kind of main neurotransmitter system that this kind of glycosylation also influences is within the kind of glutamate system. And again, it seems to tune all of the glutamatergic receptor systems. And again, as we know, there are a whole bunch of people with psychotic illness who just don't seem to respond to our kind of current treatments. And one of the reasons for that may be that they're all trying to work in the same way by trying to kind of affect the change in dopaminergic tone. And we know that works for some patients, but for about a third of those folk, it doesn't work. And for those, the more recent research suggests that perhaps the problem is not so much in these kind of dopaminergic areas, but perhaps in kind of glutamatergic neurotransmission. And over the last 8-10 years, there have been various reports showing that some glutamatergic agents reduce psychotic symptoms, kind of work in schizophrenia and psychosis. And the stage where they've tended to fail is when they go into very large-scale trials. And at the moment, there are at least 2 compounds which have shown kind of effect in smaller, a few hundred size samples. And the question will be whether they can show the same effect when they go into larger kind of production type clinical studies. The other areas where the paper makes a case for a kind of intermediate kind of brain modulation type thing is around the area of complement, which I'm not going to talk about, but one of the things that comes out as one of the strongest genetic signals is in this area of changing of complement in brains of patients with schizophrenia, and that is thought to influence synaptic change and plasticity and pruning in particular, which has been shown to be abnormal in patients with schizophrenia and is thought to be the reason why the illness kind of comes on in adolescence when there's a lot of brain pruning going on. And the final, the kind of fourth aspect, which they use to link the kind of gene to function and the role of glycosylation, has really been around calcium channel modulation. So what, as Derek said, when you add these sugars, when you glycosylate various structures, including these calcium channels, and they seem to be intrinsic to a whole bunch of brain functions, but also cardiac functions. And influencing those channels will then have downstream effects on a whole bunch of neurotransmitters, including dopamine and including glutamate. So that's an area where there has been much less interest. We have a lot of drugs which influence calcium channels, but all of them predominantly have been in the area of cardiac disease. So a lot of kind of nifedipine, diltiazem, calcium channel blockers, they're actually known as that type. And there have been trials to see whether they will also influence brain activity, but there haven't been any large-scale studies which have shown success. But what this paper suggests is that maybe we've been overly focused on the cardiac versions of these calcium channels, and actually we need to be focused a little bit more on the brain end of those calcium channels, because they influence all kinds of neurotransmission. And if you believe in that view that the neurotransmission within networks in the brain influences your kind of cognition in terms of how you perform, but also in terms of how you're making sense of the world in terms of your kind of perceptual reality. So for me, it offers quite a strong way forward in terms of how you might translate the kind of findings from this kind of approach into treatment for a disease where we don't really have very good treatment, or where they're definitely suboptimal. I don't know what you thought, Dan.
Yeah, I'm quite out of my depth with this paper, so I was kind of interested to learn from you two what you thought. I guess, I mean, to add a slightly, I don't know, a critical slant on it, I think one of the things that's been disappointing with a lot of biology, particularly, you know, genomic work, has been, and I think this is a criticism that's it's certainly not, you know, my own thoughts and ideas. It's stuff that I've inherited from other people, but there's been a tendency to jump. This, what Derek described as a Mendelian type approach, where when someone finds something on the genome, there's a tendency to hope that it maps one-to-one straight into the phenomenological aspects of a disorder as complex as, say, schizophrenia. So the idea is, you know, I've found this particular, it would be great if we could find an SNP that maps directly to, oh, well, you're going to experience hallucinations and it's something that is either treatable or preventable. I wonder, is glycosylation the new methylation? Is it one of those situations where, you know, it's another candidate post-transcription mechanism that we hope will map one-to-one onto the phenomenology of illness. But I don't have an answer to that. That was that's my critical half of my brain thinking, oh, maybe this is just more of the same in that respect. What was different? You seem to be saying, Sukhi, that the difference is that it speaks to our current understanding at the higher level, neurochemical process level. You seem to be saying that actually it maps into that quite well and marries with top-down experimental studies where people have tried, probably almost out of desperation, repurposing calcium channel blockers, glutamatergic agents. I mean, is that why you were more excited by this than, say, yet another methylation study?
Yeah, I think part of the reason is really to see, well, one, you can just become bored by reading yet another genetic study which won't be replicated and doesn't offer you a clean translational product. And I think that's kind of lazy and very easy to do, because that means you can just ignore 50% of the studies that come out on genetics, because you don't need to think about that. But I think the other way of looking at it is that they are telling you something. And over the last 20 years, they've told you that that model that we're describing with the P's and Mendel and one gene mapping onto one particular function, either brain function or mapping onto one symptom profile, is definitely not true. So everybody's thinking needs to move on. So maybe the three of us are just too old, because when we went to school, that was the model that was being sold, the new model about really subtle effects, lots of different genes interplaying, and some of them not even coming into work unless certain environmental things are there. So actually just looking at the genes in themselves is just not quite enough anymore. The effects are much more subtle. So then I kind of think this model offers a way of squaring that circle, because rather than saying it's not offering us one gene, one product, one illness model, what you're realizing is there must be some intermediary. And one of the things that I liked about this, and you're right to say that it looks a bit like methylation because it kind of affects lots of genes, so it could be applicable to everything, is it offers one kind of translational model that you can actually test. The thing I liked about this, as opposed to the methylation probably, is that the things that it linked to, so while methylation seemed to be a story which linked to every single gene in order to produce protein had to be methylated, so you could look at the active ones through methylation, this seemed to be offering an opportunity where you could look at a process which was actively modulating particular receptors or particular neurotransmitter systems. And so therefore, it kind of said, okay, yeah, that makes sense of the fact that maybe dopamine may be something that we could look at therapeutically. It kind of makes sense that glutamate may be something that we could look at therapeutically. It may be that we need to revisit the kind of calcium channel antagonist. And the thing for me is that the basic science is progressing at such a fast pace. And, maybe this COVID vaccine is a way of showing you that something 20 years ago would have taken 10 years to produce, as now you can turn that around with the modern biological techniques to something that you can produce and test in relatively short space of time. That if you say that we say that we're interested in these kind of calcium channel modulation through glycosylation, actually the basic scientists may already have some of those tools to test that and try that out. So I think that link across 3 or 4 systems, but equally the weakness in the system is exactly the same as you highlight in terms of methylation, that actually the glycosylation also influences these calcium channels. These calcium channels actually don't just selectively influence dopamine and glutamate, the things that we think are more affected, but they also influence acetylcholine and noradrenaline and norepinephrine. So it's not as clean as you would like it, but it offers one avenue, I think, which uses that genetic research in a positive way.
Yeah, I thought that was interesting. That again, it goes back to a spectrum model, and that there could be lots of different things going on, to different effects in different people. But you're right; it's so broad. The authors have a phrase about. They don't oversell it. I mean, they talk about it opens up therapeutic potentials, but it's a bit of a throwaway at the end. I think the phrase was small molecule pharmaceuticals might help, which they might. But you can see, again, if you're in any interference with this system, that has much potential to do harm. Okay, so we've generally been fans with some skepticism. I thought it was quite good. I think a lot of GWAS stuff leaves me quite cold, and I came out of this one a little bit more warmed.
One thing that reminded me when Dan was talking about the fact that, you know, you're seeking this very simplistic notion from some kind of genetic or brain change, one regional change, mapping that onto one function and then one behavior. It really reminded me of the early days, similarly about neuroimaging, where, you know, our neurological model was, well, you have this kind of Broca's aphasia. So if you have damage to the kind of left inferior frontal operculum, then that will will interfere with your speech production, perhaps. And then if it was a bit further back and it kind of affected the Wernicke's area, maybe that would be much more likely to influence your speech perception. So those early studies kind of set the ground for us thinking that one bit of the brain is going to be doing one thing which is measurable, and that causes a particular dysfunction. And what we've learned, again, kind of, you could almost say it's analogous to the genetic studies, is actually we think the brain works within networks, that a lot of it is working together in order to be able to do these things, and it has a bunch of redundancy in some areas where you can have some damage, but it will kind of be able to function using other nodes, and in other critical areas it may have less kind of surplus. I just wonder, so in a way, the maths of it, in terms of trying to find one concordance between one kind of region or lesion or genetic change to the behavior, I think has shifted. And I think that brings us quite nicely onto some of the things that were in what are the kind of 50 most progressive things that have happened in maths or stats over the last, 50 years, the thing that Dan was going to talk a little bit about. Because having all of that 20,000 voxel data, or having this GWAS data over 77,000 people over however many million genetic pairs, requires a new kind of thinking, because we can't do those simple tests which kind of compare two groups on one thing and says this is what the significance level is at P05. So I've learned a lot from Dan about this. I'm really pleased. I'm really looking forward to hearing what he's going to say. I hope he talks about the bootstrapping story.
I don't think you will follow on from the bootstrapping story. I'm not sure we will have time to fit that in. So that's onto your paper, Dan. 50 statistical terms to learn before you die, or the top 50 ideas of last year, or top 3 ideas of the last 50 years.
It's good to see that all three of us are completely on top of our respective contributions today. So it got to the end of the year, and it occurred to me that every year, there's the top X of Y list, the best films of the year or whatever. And two people that I have a real admiration for, Andrew Gelman and Vehtari, put this paper up on arXiv about what they consider to be the most important statistical ideas from the last 50 years. And because I know of their work, I wouldn't pretend to understand it, but I know their work. I thought this is going to be worth a read, right? And the reason I thought it was relevant to our discussion is because very often in the column, as well as on these events, we talk about papers where there's tons and tons of data that's generated in really high throughput fashion. I mean, GWAS is a great example, but the example Sukhi gave us was the one that ironically him and I first met over, was the analysis of functional MRI data. And the story was that I think the first meeting Sukhi and I had professionally was when I was trying to persuade him that a bootstrapping technique wasn't faking data. Because we were looking at some psychometric data, and I was saying, why don't we bootstrap it? Because we've got such low sample sizes. We can only really sample from a few people, because the experiment required them to sit through about 4 hours of doing the same task over and over again for thousands of trials. And people just won't tolerate it. So that was our introduction professionally, but and I've never forgotten. So when this paper came out and I read that bootstrapping was one of the top 50 ideas, the top 8 ideas from the last 50 years, it kind of reminded me of that story, and I thought we had to talk about it. But anyway, the paper talks about the confluence of thinking in and around statistics, and I guess the headline from the perspective of its applicability to our discussion is that in the world of big data, where for one person, an fMRI study will typically reveal or give you data where you have of the order 10 to the power 5, 10 to the power 6 data points just for a single subject. And what you want to do is establish whether or not experimental condition A or B influences any one of those possible tens of thousands of possible data points. Or given that, you're in a situation where, first of all, you're never going to be able to do that by hand, right? You'd never be able to analyze that volume of data the way you would have done in the 1940s or 50s by writing up tables and manually calculating with a slide rule. I mean, that's never going to happen. So it kind of fascinates me that the way we think about data, we think about experiments and what we consider to be valid scientific inferences, intricately connected to the development in other fields of engineering, mathematics, computer science, and applied statistics. So this paper was really nice for that, because not only does it talk about these 8 great ideas, but it sort of winds it all up and puts it together. I mean, the last part of the paper is about what does it all mean together, and there are a few things that came out of that I thought were fascinating. I mean, the first thing was when Sukhi and I first started working together. In fact, Derek, you and me also, we would never do anything unless we could do a hypothesis test on it. So it was like, we look at the graph of the data and it looks kind of like there's a difference in effects between groups. And then the first question would be, but what's the significance? How do we prove that? So there was this not trusting our eyesight. We wouldn't trust graphs of small sample sizes, but we would trust an arbitrarily defined, independent samples t-test or something like that. So we could scurry off and fire up SPSS and then run a test and then convince ourselves we found an effect because the p-value is just less than 0.05. And one of the things that comes out of this paper is, because we can now you can do in a matter of seconds with even a modest computer, the statistics that you can do now are so computationally intensive that it would take a team of, I'm picking numbers out of the air, but tens of thousands of people working simultaneously. You can do that in 10 seconds on a modest laptop. And in doing so, we're now in a position where we've moved away from arbitrary testing of small data samples, and we'll be, one of the key points in this paper is exploratory analysis using visual methods. So statistical graphing is now a thing, right? And there's even authors that are saying, I don't want to hear about your table 1 qualitative or quantitative group differences between your 2 samples. I want you to just show me distributions of those that you can generate, again, in a split second using a statistics package. So there's this move away from arbitrary and a comfort in thinking that a formal statistical test is actually robust and valid, to thinking, well, you can leverage the power of the power of the computational machinery you have, and you can actually trust your eyesight again. You can use graphs to say, oh, look, here's a graph of that data set, here's a graph of that data set. Because we can do that so easily, we can now start to use that as a formal method. So that really, I thought that was really interesting, because I never used to trust graphics. I used to trust, if it wasn't a formal test, I wouldn't believe it. But actually, that's particularly pertinent. And the other really important thing in there was the idea of regularization and over-parameterized models. And this speaks directly to what we were talking about with, fMRI data, where you've got hundreds of thousands of potential candidate model variates, right? But what you want to do is fit a model that tells you which of those is important. So you've tend to got in a typical GWAS study, you might have 10,000 people, but more often than not, you're going to have, it's going to be the order of 100 or hundreds. And the same with fMRI. It's very rare to see data sets bigger than that. And yet, for each participant, you're fitting a model that's got tens of thousands of parameters. So you've got way more parameters to fit than you have data points, right? So, traditionally, statistics would say you're onto a loser, you can't do that. But this idea of over-parameterized models and then restricting how you find those parameters, using clever algorithms to find those parameters, has meant now you can actually do a GWAS study where you've got potentially 1 million possible parameters, and you need to hone it down from a relatively small sample size in comparison. And again, that comes back to the start of the point, which was you can't do that by hand, you have to do it with a computer. So I think the thing that I liked about this paper is it's a bit like a Wikipedia page. You can read any one sentence in it and you can go, oh, that paper looks like I should have read it, and then go and read that, and then you go down a rabbit hole. So it's like a history of modern statistics. So I thought it was really, really exciting and interesting for that reason. And like I said, the reason I brought it up was because it reminded me of that very first discussion where I had to try and persuade an esteemed colleague that bootstrapping wasn't just making up data.
Well, I've paid a small child to ring Sukhi's doorbell at 5:30, so we know when the session is over, and he's just done that. But I don't know if we go to the esteemed colleague for the last 30 seconds of conclusion. I found that very convincing from you. Have you any final thoughts on Sukhi?
Not really. I think I'm convinced now that bootstrapping is not just that's the nice thing about hanging out with somebody smart for 20 odd years is that you learn lots and lots of things. So
that's all it took, 2 decades.
That's definitely sold me on reading that paper and just picking one of those papers, one a week. That'll take you through the whole year. Got 50.
Absolutely.
Well, we'll wind it up there. We hope to have Dawn with us next month, and we hope she gets well soon. But thank you for joining us.